Hi, I am Gianna Shiv, and today we will be discussing the interdependency of the cell cycle and cancer. I'm going to let my other group members introduce themselves.
Hello, I'm Catrice Cook.
Hi, my name is Ashley Gill.
Hi, I'm Chanel Carrington.
Hi, I'm Hillary Mosley.
Okay, so today we're going to discuss the interdependency of the cell cycle, cancer, and your genetic makeup. As we all know, cancer is a very important health disease in the world and probably right in our backyards with our own family members. It has been estimated that 1,600 people will die from cancer per day in 2014. And to begin to understand cancer, it's important to understand the cell cycle, the factors that regulate the cell cycle, and the effects when those factors work improperly. We will begin by discussing the cell cycle, and it is important to note that the cell cycle is initiated originally by the signaling pathway MAP kinase. And we will discuss this a little later, but first, Chanel is going to discuss the phases of the cell cycle.
What is the cell cycle? The cell cycle is a series where the cell undergoes through division and replication. The cell cycle begins with two new cells are produced by division of a single parent cell where the cell ends and one of these cells divides again to separate into two cells. Next slide. The four phases of the cell cycle. The first phase is G1 phase. It's the longest phase of interphase and occurs before DNA synthesis. G phase is in charge of making a very important decision in the cell cycle. It is charged of determining whether the cell can be able to divide again. S phase. S phase is the phase when DNA replication begins. The purpose of this process is to produce two similar daughter cells. Throughout the phase, the cell grows and continues to grow through S phase, as well as the rate of the synthesis of a number of proteins and enzymes that are involved in DNA synthesis. G2 phase, which occurs after DNA synthesis. In G2 phase, all of the organelles inside the cell are replicated in order to prepare of mitosis. Cells have one last chance to produce more proteins in G2 phase before division. Next is the M phase. M phase is called the mitotic phase, which consists of two parts. Is the mitosis phase and cytokinesis phase. The result of the M phase is two daughter cells. The first part of the M phase is mitosis, which includes the five phases: prophase, prometaphase, metaphase, anaphase, and telophase. Telophase is immediately followed by cytokinesis. In prophase, chromosome material are condensed to form a mitotic chromosome. This is assembled by the cytoskeleton. Mitotic spindles get assembled and the nuclear envelopes disperse. In the prophase, there are 92 chromatids and 46 chromosomes, and 23 in each pair. Next, there is prometaphase, where the chromosomes are being attached. When the cell enters prometaphase, its nuclear envelopes break down, and the chromosomes are attached to spindles, microtubules, and move to spindle equator. Once the chromosomes are moved to the spindle microtubules on each equator, the cell enters metaphase. After that is metaphase, where there are 46 chromosomes and 92 chromatids. In metaphase, there are fully condensed chromosomes that are aligned at the metaphase plate attached by chromosomal microtubules to both poles. Beside that is anaphase, which are characterized by two kinds of movements, anaphase A and anaphase B. During anaphase A, chromosomes and centromeres split towards the spindle poles and concord microtubules get shorter. And in anaphase B, The poles move away from each other and microtubules lengthen. Anaphase A and anaphase B can occur at the same time. By ending of anaphase, there are 92 chromatids and 92 chromosomes. Finally, there's telophase, where there are 92 chromatids and 92 chromosomes in telophase. Once telophase chromatids are clustered at the opposite spindle poles, the chromosomes become dispersed, and the nuclear envelope assembles and organelles are formed. Telophase is complete. The cell leaves anaphase and enters cytokinesis.
Which brings us to the checkpoints in the cell cycle. It's critical to know where they are, what do they do, why are they essential, what happens if cells don't pass the checkpoints, and what happens if they do pass the checkpoint. So in the next slide, we're going to talk about what is a checkpoint. A checkpoint in the cell cycle is basically a control system that monitors the cell cycle. Leland Hartwell and Ted Wiener proposed the theory of checkpoints because they knew that something was responsible for regulating the cell cycle. There are three main functions that the checkpoints do, depending on their location in the cell cycle. Checkpoints and their locations. The cell cycle is split into two major components, interphase and mitotic phase, called the M phase. During interphase, there are three phases: G1 phase, S phase, and G2 phase. M phase consists of four phases, which is prophase, metaphase, anaphase, telophase, and cytokinesis. Cytokinesis is not technically part of M phase, but it immediately follows telophase. So now we know where the checkpoints just messed up. Can we stop? I'm sorry. In this slide, I put there's four. There's only 3 checkpoints. So can we edit that? Okay. So can we go back? Can we edit it real quick? Sorry. I'm sorry. Okay. We gotta fix that because he's going.